Thank you for joining me today. I'll be happy to provide you with some update on the work we are doing at the University of Cincinnati toward bioprinting a model of glioblastoma. So my journey in the field of microphysiological systems started in 2014. At that time, I had the opportunity to join the lab of Donald Hingberg, and we were working as you. Sorry, I'm trying to use the pointer. Do you have it? Magda, did you bring with you? Oh, sorry. Oh, thank you. Thank you. So we were working with this now I think very famous chips that I'm not going to describe, but in details, but essentially consists in two microfluidic chambers separated by porous membrane. I've been working on different models, including an iPS-derived blob and barrier model. It was the first isogenic model derived on chip. But secondly, if you look at what happened in the course of these years, the design of this device has changed, and essentially hydrogels are taking over part of the initial PDMS structures, not just in brain models, but also in many others tissue models. And that, of course, makes sense because the hydrogels allow us to model, for instance, an aspect of tissues, including mechanics such as stiffness, as well as ACM composition that more simple systems do not allow us to do. And more recently, we are also start looking and observing the emergence of bioprinted microphysiological system, which I think start to be a little different categories because they are technically not chips, as they are not using anymore electronic chip relay technology for fabrication. And so in this case, for instance, I took this image from a recent paper or recent review of 2021, in which the author track the number of publications that use bioprinting approach toward generating of this microphysiological system. And for the one of you that may be more familiar with 3D bioprinting, that you may be aware that in the field of microphysiological system, at least, extrusion-assist bioprinting and stereolithography are the main 3D printing methods used for generating these systems. They're different because if you look at, at the pros and cons of the two methods, in a way, those two technologies are in a way complementary, but absolutely different. The nozzle-based extrusion bioprinting essentially consists in extruding hydrogels with or without cells to generate 3D structures. The methods, however, are relatively slow in respect to what you can do with stereolithography, in which you can have 3D printed devices that are that can have a much higher resolution, but unfortunately they usually have low biocompatibility because these resins tend to be toxic to cells. And so in the field, a number of people tend to use this one-step bioprinting approach, which is extremely elegant, and let's say we will call it a very sexy approach to generate these models. But it's also extremely useful, if you will, because as you can see in this example here on the left, what I took an example of a microfluidic model of liver that has been generated with a one bioprinting approach. The authors managed to demonstrate that they can use PLCs instead of PDMS to generate the scaffold of the system, of the microfluidic system, and input inside hydrogel-loaded cells. With the different kind of liver-specific cells, and they also shown that PLC, for instance, doesn't give the same absorption problems of PDMS. However, for instance, this system is not transparently clear. It's pretty hard to look through in phase contrast because the material is not transparent as PDMS, for instance. On the other example here, in the lab of Jennifer Lewis, also the VIS, they managed to show that it's possible to bioprint in one single step a complex vascularized model of kidney in which, however, the hydrogel is now printed inside, again, a PDMS structure. But in general, I think that organ-on-chip technology and bioprinting can be synergistically used to generate more complex systems, although there are a few areas of improvement that I just summarize in these slides for obvious reason of times. For instance, not all the models right now allow us to flow a vascularized compartment, simply because it's not that easy to actually generate a vascularized and perfusible compartment with 3D printing. And so I think there are multiple steps that need to be taken in order to democratize a little bit more these approaches and make it more amenable for large-scale manufacturing, for instance. But this all, in general, align with the current trends that you can monitor by just using PubMed a little bit, as I used to do with my student in class. Doing some of this exercise, it's easy to see that the field of organ-on-chip is growing, but there are also different trends. The field is branching. Multiple people combine, including us, combine organ-on-chip and stem cell biology to generate more predictive models, but also models that could become new tools in personalized medicine. And in this sense, we believe that probably the 3D bioprinting model could also help generating this system outside of labs that have access to microfabrication facilities. With a 3D printed, as we do with my lab, we don't need a clean room to generate this device any longer. We just 3D print the entire system, we load it with cells, so we connect to general or affordable pumps, syringe pump or peristaltic pumps, and we work in any other biology lab. Having given this broad introduction, I'll now focus a little bit more in, as I say, in the progress made toward generating of a 3D bioprinted model of glioblastoma. I think most of you already know that, unfortunately, glioblastoma is a lethal primary brain tumor. Usually, people are diagnosed with glioblastoma through MRI scan. And this is a typical MRI scan with a patient with glioblastoma. You can see that the tumors, as indicated by the red arrows here, this white, what am I doing? This white tumoral mass here, it's really the tumor mass that grow and infiltrate a healthy tissue. The green arrows also indicate these black dark spots here. This is basically a sign of vasogenic edema. The tumor infiltrate a healthy tissue and destroy the blood vessels. So the patients will suffer from this, bleeding in the brain. And unfortunately, in most of the cases, this is untreatable. And even when it's treated with surgery and radio and chemotherapeutics, unfortunately, the tumor tend to recur, and the recurrency are untreatable, meaning the tumoral cells are no longer sensitive to the chemotherapies or other treatment. And it's clear that we have a huge translational gaps. There is no model of realistic, predictive model of glioblastoma nowadays, and those are actually data coming from clinicians. Not tissue engineers, not people with a conflict of interest like myself, obviously, because what the clinicians find out is that even drugs in clinical trial, in most of the cases, are not designed to cross the blood-brain barrier. This is their data. In 2011, 92% of the drugs that were used in clinical trial were not able to pass the blood-brain barrier. And they found out themselves later. And even today, there are not many drugs able to address or to kill the glioblastoma cancer cells, as you can see from these numbers over here. And again, the problem is actually difficult to even identify these cells in existing models. The glioblastoma cancer stem cells are basically a subpopulation of cancer cells that ensure tumor self-renewal, as it will happen in any tissue, unfortunately. But also they constitute a pool of cells that are resistant to chemotherapeutics. So you can imagine that if we were able to have a model with these cancer stem cells, we could actually try to target them as well as we may find new therapeutic opportunities. There are multiple niches, however. Glioblastoma tend to have multiple areas in which of the brain in which it can infiltrate but also generate this niche. And so each self-renewal niche is different regulated. And that's why it will be important to have all, at least these three main niches in the same model. The three niches here identified are the epoxy niche, the perivascular niche, and the so-called invasive niche. There have been multiple efforts trying to model glioblastoma in vitro, but I think, as in any area, there are still areas of improvement, as in any model. This is one of the most recent one. It's a 3D-Bioprinting model that, however, is still surrounded by PDMS A and B. The endothelial cells can be eventually co-cultured with cancer cells, but are not perfusive. So even in this model, won't allow us to study drugs that can pass the blood-brain barrier and eventually kill the cells. So in my lab, we are using a combination, as I mentioned before, of stereolithography and bioprinting to generate a scalable system. Through stereolithography, we design via CAD, and then we 3D print all these microfluidic chambers, in which then we bioprint hydrogels, including a sacrificial ink or fugitive ink that is then encapsulated in a gelma or fibrin gel. Once solidified, we can remove the fugitive ink, and we have an hollow chamber that we can further coat with our extracellular matrix protein and then with endothelial cells to obtain a full vascularized lumen. Of course, on top of it, in the hydrogel, we can add other cells, such as astrocyte or glioblastoma cells, so we can have also the stromal compartment. And all of this it's fluidically active. You can perfuse this vascular channel. I will go a little bit more in details in a moment. This is actually the design we are using. We have an inlet and one outlet. We can also have multiple inlets. Outlet is a 3D printed system. You can change any time and have a dozen of that in a few hours, essentially. Again, the hydrogel is here. It's perfusible and seated with endothelial cells, as you can see in this case. And we try multiple cell types, brain relevant, including astrocytes, endothelial cells. We seeded also single cells, single glioblastoma cancer cells, or even tumoral mass in the form of spheroids. The system is transparently clear because at the bottom we have a glass slide. You can use phase just to look at the cells, as you will do in any Petri dish or any other chips. You can use standard fluorescent dye just to look at the live dead cell ratio, fixation and post-fixation staining if you want to go in for higher resolution and look at other biomarkers are obviously possible. We, for instance, look at the availability of endothelial cells and glioblastoma cells alone in co-culture to make sure they're all alive in our conditions. We also grow brain endothelial cells, primary brain endothelial cells. They form a nice monolayer, as clear from this V-cadherin staining over here. And even human astrocyte can be cultured for more than a week. They're still alive and fine as in any other 3D culture you know. So it's easy to adjust existing protocol to this device. And this is again scalable. We can easily prepare dozens of this in one single experiment. The system, as I said, is perfusive. So what we start to do is trying to look at the barrier function of these blood-like vessels. We can stain that with WGA, which is a lectin. So that stains the lumen of the endothelium, and we perfuse it with cell culture medium containing this fluorescent dye, dextran. It's a 10 kilodalton dextran that is mostly retained within the endothelium. And as you saw also in other presentation, we can also, like in other systems, look at the barrier function, so the ability of these vessels to retain the dextran in this case in healthy conditions, so when endothelial cells are growing alone or when you add the cancer cells. And as I mentioned before, glioblastoma tend to destroy the blood vessels, generating this kind of edema phenotype also in our system as it happened in vivo. This is also it's confirmed via traditional staining for junctions such as PCAM. as you can see on the bottom. Obviously, we can also take a quantitative measurement out of the imaging. We do in this case, we just look at the amount of dextran and extra waste from the main channel, and it's easy to see that the system and the experiments are pretty reproducible. We're able to look at this vasogenic edema-like condition in vitro. And clearly, we can also perform more complex and dynamic imaging to really see how fast the dextran, let's say, extra base also with this model when in co-culture. But it's something that I found more interesting to present to you is this initial observation we made a few months ago now, we've repeated multiple times, is that if cancer cells and primary endothelial cells, so healthy cells, are co-culture in dynamic conditions, so under fluid flow, we frequently find that the cancer cells tend to have this peculiar morphology. They become essentially round, okay, which is different when you have cells alone or cancer cells co-culture but in static. The roundness and the number of cells around, here identified in yellow, are a lot more in dynamic co-culture condition. And if you look at the entire length, actually width of the chip, you see that there is actually an extreme gradient of morphology that is here. And obviously, you can also look at aggregate magnification, and we suspect that probably this is more a phenotype that you will see in a perivascular niche, round. We start looking at some specific markers of self-renewal here, and I can tell you we already found some of them. Now we're looking for a marker of this probably second niche or invasive niche. There is a lot to do, still to do, obviously, from the biological perspective. In terms of biofabrication, I think we have a solid system that allows us to produce a scalable microfluidic device with a 3D bioprinting approach, and the system can be used it's technically a PDMS-free system that can be used for culturing multiple cell types. Part of ongoing work is now to integrating also spheroid or organoid that will recapitulate better the hypoxic niche of glioblastoma and better characterize this invasive front and the perivascular niche that we probably are able to capture already. Having said that, I would like to thank Sujana Poon, who is in the audience. He's the PhD student who is carrying on most of the experiments now, and our collaborators, Spot, at Cincinnati Cancer Center and Custom, that are helping us in this process. Thank you all for your attention.